aspects of gene mapping. Okay. So can we start, Rajlakshmi? Everything okay?
Sir.
So let us see this.
Sir, yes.
Yesterday we have discussed about the gene mapping, but we didn't discuss about the what is the distance between the EGR and TAN-R. So today we will discuss the same. Now my favorite screen black. Now you can see. Suppose we want to define, because meiosis is absent in the bacteria, right? So we have to adopt the different strategy. Okay. So suppose this is a time against percent co-transcription or kind of conjugation or co-transformation, whatever the term you would like to use. Suppose this is the gene A with time 5 minutes, and then gene B 15 minutes, and gene C 20 minutes, and gene D 25 minutes. And there are 4 genes A, B, C, D, right? So I am giving the very simple example so that you can understand what I am talking. So distance between A to B is how much? Can you tell me? And between B to C, how much? C to D, how much? B to D, how much? A to D, how much? From this graph, what we can say?
The distance between A to B is 10 minutes, sir.
A to B is 10 minutes. Okay.
10 minutes.
Means 15 minus 5.
B to C is 5 minutes.
Okay. Very good. 20 minus 15 it will be 5
B to C is 5 minutes.
C to D.
C to D is also 5 minutes.
Right.
25 minus 20 is also 5 minutes.
B to D.
B to D is 10 minutes.
Okay.
A to D is 20 minutes.
25 minus 15. Okay. And A to D is 25 minus 5.
20 minutes.
And so I think this is very clear to you, right? So if such kind of a graph you can see in the examination, then automatically you can calculate what is the typical distance between EGR and TAN-R. So it's a very close, so they will not ask you, but certainly they can ask you in between this, any kind of question they can frame up. This is the percent frequency of HFR, and this is the graph, appearance of donor genetic marker in recipient as function of time, and how you will calculate the distance. Because E. coli, if you will see, the E. coli mapping, and this was the complete mapping called as the 100 minutes. Okay, so only the time frame, and yesterday I have discussed you that 1 minute is equal to 1 unit. Okay, so because meiosis is there, so in eukaryote we can easily see the meiosis, so we can determine the recombinant frequency, non-recombinant, double crossover, SU. Do you know about the single crossover and double crossover? So yesterday we have discussed this, right? And by the transfer function experiment, we will take complete map. Okay, so And I was discussing about the interrupted mating, that is co-transduction between when the viruses is going to attack the bacteria, and then how we can calculate the interrupted mating between the bacteria, between the viruses, and actually the timeline is there, and which is the equivalent to distance unit. So now I think you can solve this problem. Okay, so you can see this graph, and based on this graph they can ask you design the map. So you can draw this map. For example, this was at 10 minutes there were 2 genes, AZR and AZ done, and then at 15 minutes exactly this was a lag, and then between 15 and 20 this was a gal plus. So easily you can design a map, right? And then actually map distance, and just now we have discussed a unit, 1 minute is equal to 1 unit. So this is the 100 minutes E. coli complete map. You can see this is of how much minutes? 100 minutes. Okay, and yesterday we have discussed all these things. And also we have discussed this point, okay, how you can combine and you can find out the distance, or you can design the map based on the direction of origin, because linear chromosome is absent in the bacteria, always they are circular. Okay, so that's why you have to think in that way. So now we have yesterday we have discussed how the T4 bacteriophage is infecting E. coli and how we can go for the gene mapping. And yesterday also discussed about the lytic mechanism, lysogenic mechanism, and based on their feature we can map the presence or absence of gene. For example, if there is a lytic cycle, then all the cell is going to be dead. Cell is going to burst. And if there is a specific if there is a specific transaction, suppose a recombination between the phages gene and bacterial gene, then another cycle can operate, that is called the lysogenic, right? Lysogenic cycle. So based on this feature, we can now we are in the situation to map the gene, and that's what was the basis of Benzer to define the gene based on this packaging, based on the lytic feature, based on this lysogenic feature. For example, suppose this is the phage attacking the bacteria, right? And one of the genes C plus is going to be inserted upon the infection. Then what next? Phage replicate, own DNA, make protein head, right? Host cell degrade, the host chromosome is cut, gene C plus is present on DNA fragments, okay? And C plus is packaged instead of phage DNA in one of the 1,000 of new phages, okay? So phage particle with C plus move to another host. So what you can finally observe that the packaging is done that was introduced, C plus. So that is a novel feature. You can see inserted in genome via double crossover. So crossover recognition is also part of bacterial. So you can easily map based on this feature. Okay, so bacteriophage. Now in the lysogenic phase, what you can observe, circular phage chromosome, and there is a recombination between the bacterial chromosome and viruses, that is forming the prophage, and In this way, they are producing the lysogeny bacteria. I think this is clear. This is clear or not?
Yes, sir.
Yeah. So this is the basic feature of mapping integration of viral DNA into the bacterial genome. A virus that can integrate what you can call them as a template and integrated phase is termed as prophase. So non-violent units that are inserted in the host chromosome and multiply by binary fission along with the host DNA. So prophase has a feature that they can enter the lytic cycle to complete the virus life cycle. But this induction is based on certain either external signals, for example UV rays or any other kind of induction. So prophase expresses a regressor protein that inhibits further infections. So this is the feature of what you can draw from the life cycle of bacteriophage over the E. coli, and also this inhibitor prophase DNA excision gene and gene used during the lytic cycle. So automatically between the two cycles there is a control, that is a regressor, right? And certain environmental cue that I was discussing, they can also block the expression of a regressor protein. For example, if the DNA is damaged, then this repressor protein expressed may not be expressed. So prophase excises and enters the lytic cycle. So you have to know this feature. If you are knowing this feature, then further you can predict about the mapping, gene mapping, and their other cycles. Okay. So that's why you will see there is a specialized transaction, and because in this event everything matters, like repressor, activation of repressor or certain environmental signals. So what happens upon excision of prophase, the adjacent host DNA is taken along, the completion of the lytic cycle and subsequent infection of another host moves the flanking DNA to another bacteria. For example, you can see in this excision, what you can observe, that this is the AT feature, alright. AT is present in both this lambda DNA and E. coli chromosome. So that's why they can cross over at this point. And if they can cross over at this point, then you can see easily there is insertion of AT gene between the gal and bio, right? And then this lambda DNA is excised. So resulting phase DNA and E. coli chromosome has a separate feature after the recombination. Recombination is there. Recombination is because of certain common sequence. For example, we are saying ATT is common to both. Okay. Now, now you can see what is the abnormal part if gal is going to be part of the lambda DNA, right? So forming the lambda DG DNA. So that's why we are talking about the normal transaction and specialized transaction. So in a special condition, something different is going to happen, right? So yesterday I was discussing about this feature, how you can prepare a transaction mapping of bacterial chromosome based on the certain markers. Okay. So I will discuss later on, but first we should know about what is a specialized transaction. Specialized transaction is used for moving specific genes between bacteria, but not for general genetic mapping. So what are the features? Because certain time CSIR is going to ask certain features. What are the features of a specialized transaction? Which is true, which is not true. So you must know this feature. Alright. So specialized, as you know, this is the term specialized. So this is really specialized, means excision is very precise. And rarely excision results in genetic exchange with a fragment of lambda DNA remaining in the E. coli chromosome and some bacterial DNA added to the lambda chromosome. And third point, resulting transgenic phase is designated as a special name also if there is a transaction of GAL plus and that is added to the lambda chromosome. So now this lambda DNA will not be lambda DNA, it will be lambda D GAL plus D for defective. So it's not all for gene alone. And lambda D GAL plus can replicate and lyse the host cell since all lambda genes are present either in the phase of bacterial chromosome. So that is the part of specialized transaction. It may be any gene, but the common feature is this integration is because of at site. At sometime at are present in E. coli chromosome as well as bacterial chromosome. Right. So you can see the production of lysogene, circularized phase lambda chromosome, and they are meeting this common junction at ATT. What you can see, there is a recombination, right? Region of homology exists as a ATT in both the bacterial as well as the lambda chromosome. So now you can see they are integrating and making a common chromosome. That may be the one condition, and other condition may be they are normal outlooping at a different place, and then there may be excision and excision with the genes. That may be one condition, and another condition may be a rare abnormal outlooping due to which you can see the galactose plus is going to be inserted in the lambda chromosome. So these are the normal feature. Lysogenic formation of lysogenic transductant and transductant produced by recombination, and then you can see different kind of transductant you can obtain based on the recombination feature. So in examination they can give you the different conditions. So you have to look for the homology where they can recombine, or they are combining because of abnormal condition at abnormal place, what they are exchanging, and they can ask you the final product. So what will be the final product after the recombination? For example, here you can see lambda GAL plus Fas chromosome is combining with the normal bacterial chromosome. Then what is happening? You can see the resultant lambda GAL Fas chromosome unable to replicate because not all genes are present. So, but another way in the bacteria you can see stable GAL plus transductant, right? So this may be the different condition. Transduction of GAL bacteria by initial lysate consisting of lambda and lambda GAL. These may be different conditions that can be asked. If they are asking about the mapping of the gene, that will be very simple question, but you have to identify what is the condition. So here you can see this is bacteria and co-infected by two different parental phases. One is the H plus and H plus R, and another one is HR plus. Okay, HR plus. So now what we are expecting, a replication of Fas chromosome in the cell. So both gene will be inserted, right? Both gene will be inserted, H plus R and HR plus. Okay, and then H plus R, H plus R, H plus R here, replication of Fas chromosome in the cell, and this HR plus, HR plus, HR plus. Then in the next page, what you can see, some are recombining, some are not recombining, right? So if they are recombining H plus R and HR plus, what you can expect? You can expect parental as well as recombinant. This is clear? So this is the basic fundamental aspects of gene mapping. Always you have to see the recombinants, right? And based on the recombinants, you can find the distance between the gene, order of the gene, how they are behaving, because based on the parental you have to compare with the parents. So what we can see, parental will be the as common as we have taken H plus R, or either or HR plus. Now you can see there are two parental HR plus and two parental H plus R. In the recombinant, what you can see, H plus R plus, and another will be HR. Clear? This is clear or not? Okay. So then Benzer did this fine structure analysis of bacteriophages, and based on this they started intragenic mapping, and also this was showing the mutation site within the genes. Okay. So and the basis of this, and they define this as a complementation test, Strong's behavior, the complementation. What is the complementation test? Complementation test determine how many genes are involved in set of mutation that produces a given phenotype. For example, what are the example that was taken by the Benzer was T4R second, right? T4R second, and there was two gene R second A and R second B. So they can have the two different phenotype if the one mutation is there here and one mutation existed here. So what will be their phenotypic appearance? Phenotypic appearance is the formation of plaques. Plaques is halorism, where suppose you are growing a lot of bacteria, right? And what is the nature of bacteriophage? They kill the bacteria. So you can see the halorism or plaque region. Okay. So you can see the clear plaque, right? Clear plaque. If they are killing, if they are not killing, you cannot see any kind of plaque. Based on the plaque morphology, based on the plaque morphology, you can see, you can identify the difference between the plaque मतलब हैलो, जहाँ पे किलिंग हो गई, सारे बैक्टीरिया को किल कर दिया, खाली स्थान बन गया. That is called as the plaque. Plaque is the empty place where all the killed bacteria. Bacteriophage eat bacteria, na? Each bacteria means they kill bacteria. Okay. So there is no bacteria you can see at this region. Virus has killed all the bacteria because of lytic cycle. Okay. And on the basis of complementation test, Strong's test, Benzer identified that there is a gene responsible for this behavior. Sometimes they are getting the plaque, sometimes they are not getting the plaque. So what he did, he developed two strains and he mixed two strains. One was the non-permissive strain K12, and then infected with R second mutant, and then he observed certain points during the experiment. So what he observed, if progeny are produced, the two mutants have complemented. If progeny are produced, two mutants have complemented each other, and that's why their gene were complete. Gene were complete means H plus R. This was the one gene, and the gene was HR plus. So if they are making the recombinants, then only they can show their morphology. They can show their behavior as a H plus R plus HR. Okay, HR. ठीक, इतना clear है? H plus R plus HR. So what you can see, this is a kind of complementation. Neither this can grow, neither this can grow. So this is there is a gene that is involved for the responsible for the production of progeny. So if protein is produced, two mutants have complemented each other by providing different gene function, either by genetic recombination producing a few plots or complementation. If complementation is there, then complete lysing will be there. So complete plot, few plot, मतलब partial plot, है ना? तो अगर genetic recombination हो रही है तो chances are there they can recombine or they cannot recombine. But if they are complementing, certainly one dominant gene is there, and they are going to give you complete plot. So in fact, bacterium with two false genome, genotype of 1 is R second A plus. R second A plus, always you have to note down the genotype because gene is there. Huh? So always A plus is the gene. R second is the strain. Okay. So R second A plus and then R second B. And another combination was R second A, R second A oblique R second B plus. So this was the first combination, R second A plus and R second B, right? And one first providing the R second A product, the other providing the R second B, and then you can see here you can obtain it this kind of product. Okay, so now what you can expect? Complete lytic cycle. So if complete lytic cycle is there, then what it indicates? Complementation or recombination? This will be complementation. This will be complementation. Okay, this will be what? Complementation. So if no progeny are produced, both mutations are in the same functional product, DNA HR. What you can see, this is a cis, right? And this is in the trans position. See, both the alleles are HR. So what you can see, you can not see any kind of product. So that's why this was also called as the cis-trans test. Cis-trans test, right? इतना समझ में आ रहा? Is this clear?
Yes.
It's a very simple concept. If the gene are in the same, same place, same gene. For example, A and B, these are the mutants, and both the mutants are at a same linear gene. So they are going to be called as cis. And if A minus and B plus, yani B plus is in the trans position. So position matters. What Benza's conclusion was that the first thing, position of the gene matters. If they are in the cis phase, they are not going to produce any kind of progeny. So for the HR, no progeny, right? H plus R plus they can produce the progeny, but HR alone cannot produce the progeny because both the mutant are a same functional unit. So that's why they define later on cistron, recon, muton. Okay. So Benja work show that two functional unit for the R second phenotype, complementation group R second A, R second B, and both gene product must produce for lytic cycle to occur. Allele may be arranged two different way in the cis-trans complementation experiment. When the mutant allele are on two different chromosomes, as in the complementation experiment work, they are in the trans calculation. So I think this concept is clear. What is cis? Same functional unit. Okay, and trans, different functional unit. So which can show the complementation effect? If they are in the trans, right? If they are in the trans, for example, this. right? The transposition, both for both, this is transposition. They can show the complementation.
तो नॉन एलिलिक पे रहेगा तो कॉम्प्लीमेंटेशन होगा,
हाँ
मतलब वही ट्रांस।
हाँ, इसमें कॉम्प्लीमेंटेशन क्या शो होगा? नो कॉम्प्लीमेंटेशन। तो क्वेश्चन फ्रेम कर सकते हैं। बेंजर का बहुत ही पुराना एक्सपेरिमेंट है, तो बेस्ड ऑन दैट ओल्ड एक्सपेरिमेंट दे कैन डिजाइन अ न्यू क्वेश्चंस एंड दे कैन आस्क
इसमें
आउट ऑफ
कॉम्प्लीमेंटेशन नहीं होगा
इसमें
नो कॉम्प्लीमेंटेशन।
हाँ, नो कॉम्प्लीमेंटेशन, दिस इज यस कॉम्प्लीमेंटेशन। ओके, दैट्स व्हाई डिस्कसिंग जेनेटिक्स इज अ लिटिल बिट टफ, है ना? जनरल IQ level person cannot understand. Only very genius personality can understand. Raj Lakshmi, are you getting this? I think you are getting this.
Yes, sir.
So you can
Yes,
understand
sir.
by the graphical way also. This is the E. coli Q12 bacteria and falls with mutation in R2 A, mutation in R2 B, and then you can see R2 A and then R2 B. This is showing the defective B product, defective gene, minus gene, right? This is minus gene, absence of gene. This is plus, this is plus. Okay, so if they are showing the complementation effect, then you can see the progeny first produce the lysis of host. So based on the plaque form, you can count the plaque. Easily you can count. This is 12 or this is 30 or this is 50, right? So if more lysis is there, you can count the plaques, and based on the plaques, you can analyze what is happening internally. They are showing the complementation or not. All right. If suppose this is the defective product and this is and they are in the same site. They are in the same site. Now they are not in situation to show the complementation effect. Okay. Can they show? Can they recombine? What you will say? If R2 A, mutant one, and this R2 B, this is also mutant one. They are on the same site. Same site. Okay. So this is also kind of cis position. And they cannot recombine or can't show complementation effect. Alright, so what you can see in the form of progeny, no plaque is going to form because they cannot complement each other. So कमी तो रह गई, जो भी कमी थी वो रह गई. Whatever the lacuna was there, so they remains there. So based on that, you can calculate. Benzer called this genetic unit of function defined by cistron's complementation test as a cistron, right? So I think you are well aware of this fact. Cistron is what? The smallest segment of DNA. Smallest segment of DNA. And now this is broadly called as a G. Same thing is there. Cistron, smallest segment of DNA, and this is now modern term they are called as the G. तो वो मैं दिखा देता हूँ कि इसमें कैलकुलेशन कैसे करोगे. I have on the slide.
Yes, sir.
So हमने एक आज नया डेफिनेशन निकाला है, जेनेटिक्स इन शॉर्ट, और अपने इंस्टीट्यूट का नाम भी बदलने जा रहे हैं, ऑल इंडिया इंस्टीट्यूट ऑफ मेरिट लाइफ साइंसेस. तो अभी कर देंगे इसको. So a little bit revision. You should know if such kind of problems is given, you should how you will identify the SCO and DCO. So first you should know about the SCO and DCO. तभी आगे का काम कर पाओगे, because after that only formula is applicable. So what is the parental? They are highest in the number. Okay, and if they are the lowest in number, then for example 3 and 4, and now you cannot see the 3 and 4. All right, so they always will be called as what? Below DCO. And in between, in between you can call them as a SCO. तो अब फॉर्मूला जो बनेगा वो DCO और SCO को मिला के करना है फॉर द डिटरमाइनिंग द जीन डिस्टेंस, है ना. जैसे आप देखो 382 थे क्या, सिनाबार वेस्टिजियल, राइट, एंड नाउ यू कैन सी सिनाबार रूफ वेस्टिजियल. 3 अगर इफ दे कैन आस्क यू व्हाट इज द डिस्टेंस बिटवीन सिनाबार एंड वेस्टिजियल, राइट, then what you will do, you have to apply the formula only. What is the formula? To determine the map distance or percent recombination, you have to see what is the number of total SCO phenotype between the gene, specific gene, and what is the total number of DCO. Okay, and then total number of progeny into 100. So you have to include both SCO as well as a DCO. SCO is the single crossover, DCO is the double crossover. आपको छोड़ना नहीं है, ठीक है ना. जैसे सिनाबार, सिनाबार कहाँ पे है, यहाँ पे है, यहाँ पे है, सिनाबार यहाँ पे है, ठीक है ना. तो कितना हो गया SCO, SCO ले लो 44 हो गया प्लस 59, ठीक है ना. ये क्या हो गया, what is this? This is a SCO. So what will be the final addition? 103, I think, right? And what about the DCO? DCO will be how much? 3, right? So total will be 106. And what is the total now? Suppose this is around 1000. Okay, then multiply by 100, and now you can calculate. This is around 10.6, है ना. So now you can calculate the distance between Cinnabar and Vestigial. Alright, so similarly we were calculating about the colony forming unit because bacteria don't show the meiosis, and Benger's experiment I have explained. So what formula you can apply to determine the percent recombination frequency in that experiment where phases are there, bacteria is there. So this will be the 2 multiplied by number of what? Plaques. 2 into number of plaques upon this will be plaques on K12 lambda upon number of plaques on B. Okay, number of plaques on B. So what is the smallest unit of recombination detectable? Suppose one mutation is there in 10 power 9 progeny. ऐसे भी क्वेश्चन पूछ सकते हैं. Suppose there is one mutation in 10 power 9 progeny. So what will be the RF value? Simply multiply 1 into 10 power minus 9. Okay, simply multiply 1 into 10 power minus 9. Got the point? And suppose minimum distance cannot exceed 1 into 10 power minus 7. Okay, so then you have to adjust. What you can adjust? 1 centimorgan that have been calculated in the flies is 5 into 10 power 5 base pair. How much? 1 centimorgan in flies. Note down. This is 5 into 10 power 5 base pair. And base pair is separated by 2 into 10 power minus 6 centimorgan. So you can easily detect if there is what is the possible distance. For example, ये देखो calculation में recombination frequency 2 PFU on K, PFU on B, and suppose Benzel's recombination frequency is 0.02. So tell me, is this equivalent to 0.02 centimorgan? Yes. And 1 centimorgan is how much base? This is 100 base pair. So these are the specific information you should have, right? Because 1 centimorgan 100 base pair and 0.02 centimorgan means what? How much base pair? They can ask you. 0.02 centimorgan means how much base pair? Convert. Tell me.
20, sir.
This is 20, right? And there are, in the T4 genome after the sequencing, it has been observed that there are 160,000 base pair. So this may be equivalent to 1600 centimorgan. So easily means enough base pair are there to calculate the recombination frequency. So they can ask you such a question: okay, what is the smallest unit of recombination that can be detected? So what is the detectable recombination frequency? This much. Okay, 2 into 10 power minus 6 centimeter. Centimeter. Sorry, centimeter. Okay. ठीक है. So this is one of kind of calculation, and already we have discussed this. There is mutant 1, mutant 2, and how they are going to produce block or partial block or full block. Alright, based on the recombination, you can see them, and based on their result also, a different type of mapping was produced that was called as the deletion mapping, because mutation cannot be denied, because this is the double mutant. What is this? This is mutant 1, mutant 2, and the cell may have the gene that are double mutant. So gene may be wild type, gene may be double mutant. So wild type will prefer to grow more than the mutant. There is a few plaques or no plaques. This is a full of plaques, means this is wild type. Clear? Getting my point? So I will tell you how the deletion mapping can be done. So just for the information I am giving, this recombination was conducted by 3 different scientists in the different organisms. For example, Oliver experiment, there was a lozenge locus on Drosophila. Benzer experiment, which locus was there? R2 locus was there in the Bacteriophage T4. Complementation test, Lewis on Drosophila. Okay, complementation test, Lewis on Drosophila. So these are 3 type of different tests was performed, and all that observation was the same. So most popular was this Benzer's R2 locus gene, and just we have discussed based on the plaque morphology, right? So now you can analyze what is the permissive host. Permissive host means FOS will grow. A restrictive host means recombinants will grow. Getting my points? What is the permissive? This can be asked in exams. What is the difference between permissive and non-permissive? So permissive is R2 minus and R2 plus phases and they can grow. Right? A non-permissive or recombinant. Non-permissive, also called as recombinant. Recombinant. Non-permissive also called as a recombinant, and R second plus recombinants grow. Restrictive host. So in summary, you can see equalized chain, two type of equalized chain, B strain and K strain. What is the feature of K strain? No cloud formation, very small and ragged. Ragged means not exactly circular. And B strain, what is the feature? Large. What is large? Large plaques, rounded plaques in the R second. And in the R second plus, you can see the B and K both have the same features. Only difference was where? In the R second. Okay, in the R second. K strain was not forming any plaque, while B strain was forming large round plaque. Okay. And जितने depth में जाओगे, then you can understand more. You have to go into the depth. You have to read. You have to analyze. Analysis is the biggest part of success in the journey of life sciences. Because if life sciences people have no brain or no common sense, then it's very difficult to survive. Because life is changing, the microbes is changing, behavior is changing. So you have to be very analytical. And all those scientists that were popular and framing these experiments, and then they make their own identity. And we are very much concerned about the what career, what will be our career. So because we are struggling for our survival. So, but they were also struggling for their survival, but they were planning something different. So now I think you got the clear idea between the strain B, E. coli, and E. coli strain K, right? So this is E. coli K strain, E. coli B strain. Now what you can see, larger plaque based on the R second mutant 1, R second mutant 2, double mutant 1, 2, wild type R plus. So mutant, what you can write, R second mutant 1, red color, blue color R second mutant 2. Now red means mutant. All these are mutants. Sample progeny causes plated on ln of V, and then this is mutant 2, and if they are recombining, then they become the wild type, otherwise they become the double mutant. So what you can expect as a feature, all 4 phases type can grow in the E. coli B. Okay, and a lot of analysis were done by this venture Innova and all that. They were comparing what are the mutable sites, and then they developed this hypothesis of cystine, recon, mutant. So what we can conclude, we can learn that gene order can be determined in this transaction, that if recombination rate is high, the genes are far apart. If the recombination rate is low, genes are very close together. So recombination between 2 mutants to give a wild type form of the gene, right? So finally we have explained this, discussed this, and so based on the survival on B and K, now you can see what is the deletion mapping from here. This is the 4 plus 5. If they are recombining, they are producing 4, 5 at one locus and plus at different locus. 4, 5 is going to survive on B only because they have complemented. Right, you can see they are all survive on B only because all the genes are aligned. But if we are producing two mutants, one is 4 plus and plus 5, and both are combining, then the 4, 5 will be the growing, and this plus will be growing both on the K and B. Okay, so gene is the unit of change. They survive on B only, already discussed. And third condition will be that no recombination is possible. Then you can see both, none of will grow on the K, both can survive on B only. Based on that, they concluded the concept of deletion mapping. Deletion mapping आपको समझ में आ रहा है? Now it's easy. They can give you such a deleted region, D1, D2, D3, D4, right? And this may be the gene structure or gene number. Okay. So for example, here it was not written, so it's difficult to explain, but here it's everything is written, so you can explain. So they are making the overlapping. You can see this one is overlapping, right? This one is overlapping, and then means both are common. Getting the point? Both are the common. That's why they started this one. So what you can conclude, now you can see this is the deletion 1 from 1 to 4 we can see, right? And this was the deletion 2, 3 to 8, deletion 3 from 7 to 11, and deletion 4 was from 10 to 16, right? And if you have to mark this region as a gene A, this gene B, this gene C, this gene D, right? So easily we can make a map of A, B, C, D, and even they contain the more gene, and you can arrange all these genes as A, B, C, D, E, F, G. This is clear, Lakshmi? You can open your unmute yourself. But there is no problem. So this was a very important experiment, and you should understand complementation test and deletion mapping I have explained, formula I have explained, right? So based on that, new concept of gene was given, that is a strong recon method. And we can also discuss the transaction frequency, how to calculate. So you should have a common data in your mind that E. coli chromosome has how much base pair. This was asked directly in some examination, 10 power 4.6 into 10 power 6 base pair. And P1 Fas, how much base pair is there? 10 power 5 base pair. So as you know, after infection of E. coli, the Fas gene is replicating by rolling circle mechanism, and the Fas is packaged into Fas particle one headful at a time, right? One headful, this is very important. One headful at a time, means continuously is not there. So we can assume that 1 over 300 Fases mistakenly packaging the E. coli chromosome DNA instead of Fas DNA. Then what happens? So each Fas particle will pack about 1/50 of the E. coli chromosome according to the number. We can calculate this. By combining probability, we can see that 1 out of 15,000 Fases will carry a particular E. coli gene. What does it mean? Means transaction is there. A basic transaction experiment we can plan, and we can use this marker A and marker B as assumption. So group P1 on A plus B plus, in fact, then A minus B minus, and select for A plus and then screen for B plus. then you can calculate this recombination frequency. So this is very similar to the Benzer experiment. Okay, you can see here this is A plus B plus A minus B minus. So you can measure the frequency of co-transaction. You can measure the frequency of co-transaction of B with A, and then you can calculate the distance between B and A or A and B. If the distance between A and B is greater than 1 headful means not more than 10 power 5, then there will be no co-transduction. How much if A and B distance is more than 1 headful, that is 10 power 5 base pair, then there will be no co-transaction. If an A and B are very close together, then there will be 100% co-transaction. So we can draw a graph between the co-transaction starting from 0 to 100. So 100, what it indicates? Genes are very close, and as 1 headful is going to occur, then the genes are far apart. Okay, so the packaging is done. So just a minute. Now you can calculate the distance between G based on the infection, based on the progeny, and for example, we can use a 3-factor cross. Actually, this is a 3-test cross करते हैं. As we are doing the 3-test cross in the eukaryotic gene, so similarly in the phases, we can use a 3-factor cross on the gene order. You have to grow P1 on A plus, B plus, C plus, infect A minus, B minus, C minus, and then you can see what is going to obtain as a recombinants. Alright, so this is 2 type of recombinants, very similar to SCO and DC. So based on that, you can apply the formula, alright, and then you can calculate. So always they will give you the numbers, and then again we are starting the new mapping technique that is called as the restriction mapping, where the restriction digestion is done of the genome by the 2 different restriction enzymes. For example, EcoR1 digesting this plasmid PGN101, and this is the actual side of fragment obtained 20 kb. Okay, total length is 20 kb. And by the BAM-H1, you can see the different fragments, shorter fragments, 2 kb, 6 kb, 12 kb. And by the both the digestion, you can observe 2 kb and then 6 kb. And what is missing? That is 12 kb. So 12 kb is fragmented into 12, 8 and 4. So first you have to make the linear combination, and then you can draw it into the circular combination. Right, and then you can also locate, suppose EcoR1 20 kb, this is the total length, and BAM-H1 12, because 2 and then 6 and then 12. Okay, so this is also equivalent to 20. Getting my points? Okay, so if you are using the combination of both EcoR1 plus BAM-H1, then you are getting a new combination, that is 2 is there but 4 is also there, right? So 2 and then 4 you are getting, and then 6 you are getting, and then 8 you are getting. Alright, so what will be the gene order? Based on the BAM-H1, you can create BAM-H1, this is 2 kb, 6 kb, and then combining 4 and 8, this will be the 12 kb. Okay, right? So after the both the co-digestion of both the restriction enzyme, you are getting 2 extra fragments, that is 12 divided into 4 and 8, and that's 12. You are creating 8 here and 4 kb here. So your map is ready. Got the point, Lakshmi?
Yes, sir. Yes, sir.
This is clear now. So a lot of questions they can ask in the examination like this, but you should not be confused. You should not confuse. Use your common sense and see where there is a difference, and based on that you can prepare. There is no formula, just simple common sense. Okay, so this is the final map. This will be 8 kb, 4 kb is here, 2 kb here, 6 kb here, and if you are combining this will be 12. So you have to satisfy this condition. Means there should be 12 kb, 2 and 6 in the biomerchant digestion, and in the eco R1 it should be total 20, and in the both the co-digestion you just should have another 2 fragments, 8 and 4 kb. Similarly, you can practice. They can give you the such kind of gel also. So based on the gel also you can prepare a map like this. So what you can see, this is the size standard. You have not to count them. And this is the Eco R1 PST1. You are getting the 2 fragments. Eco R1 biomatch1. Again you are getting the 2 fragments. Eco R1 all are combined. Then you are getting the 3 fragments. Okay, so what is the size? This is 2.6 kb. So what you can now individually first draw the map. This is Eco R1 PST1. Smaller one is 0.46 and larger one is 2.14 kb. Okay, so what you can write, Eco PST1 and then Eco biomatch1. What you can see, 2.4 kb. This is a larger fragment. A smaller fragment is just half, 0.2 kb, and this is now 2.4 kb. Okay. And now 3 you are mixing, all 3 EPB. Now you are getting the 0.2 as well as 0.46 and then 1.94 kb. So after combining 0.46 and 1.94, what is coming? It's around how much? You can calculate 0.46.
2.40 plus 0.2, sir.
0.46, 0.2 and then 1.94. So you have to combine 1.94 plus 0.46. 2.4 आ रहा है ना? This is equivalent to
2.40. Yes, sir.
So
Plus 0.2.
2.4 is going to be fragmented into 1.46 and
together.
These are combining together. So now you can draw the
and 1.94.
Now you can draw the circular map easily. One will be the larger 2.4 and then 0.2. This is 0.2 kb. This is 0.46 and this is 1.94 kb. And they are satisfying your actual size. So easily you can draw the map and the location based on the electro forces. Now about the Lord score. What is the Lord score? Tomorrow we will discuss this. I think a lot of things I have to tell in the Lord score, then only you can understand. Okay? So this is the actually related to the agriculture in genetics. So tomorrow we will discuss about this and Lord score another kind of mapping. Okay? Thank you.